Hey everyone, welcome back to my channel. Let's see today what causes depression. The common misconception that depression is caused by a chemical imbalance is that the illness is more complicated than that. According to research, having too much or too little of a certain brain chemical does not cause depression. Rather, a variety of factors such as inadequate brain regulation of mood, hereditary susceptibility, and stressful life events may contribute to depression. It is thought that a combination of these factors cause depression. Chemicals play a role in this process, to be sure, but it is not as easy as one chemical being too high and another too low. Instead, a variety of substances are at play, acting both within and outside of nerve cells. The dynamic system that governs your emotions, perceptions, and life experiences is made up of millions, if not billions, of chemical interactions. With this degree of intricacy, it is easy to understand how two people could have similar depressive symptoms, but that their underlying problems and the best ways to treat them could be very different. Although scientists have discovered a great deal about the biology of depression, their knowledge remains incomplete. Important discoveries in the biology of depression include the identification of brain regions associated with depression effects, the understanding of how neurotransmitters, which are chemicals, facilitate communication between brain cells, and the understanding of the influence of genetics and lifestyle events on depression risk and symptoms. Mood and brain areas. There is a common belief that feelings are found in the heart. However, science links the brain to the location of your emotions. A few brain regions are involved in mood regulation. Researchers think that neuron cell connections, nerve cell proliferation, and nerve circuit function play a key role in depression, even more so than levels of particular brain chemicals. More advanced brain imaging techniques like functional magnetic resonance imaging, single photon emission computed tomography, and positron emission tomography allow for a far closer examination of the functioning brain than was previously feasible. For example, an fMRI scan can monitor the alterations that occur when a particular brain region reacts to different tasks. By determining the location and density of neurotransmitter receptors in specific brain regions, a PET or SPECT scan can map the structure of the brain. Thanks to the use of this technology, we now have a better knowledge of which brain regions control mood and how depression may also influence other functions like memory. The thalamus, the hippocampus, and the amygdala are regions that are important in depression. Studies indicate that certain individuals with depression have a smaller hippocampus. For instance, researchers examined 24 women with a history of depression in one fMRI study that was published in the Journal of Neuroscience. The hippocampal size of women with depression was on average 9 to 13% less than that of women without depression. The woman's hippocampal size decreased with the number of her depressive episodes. Experts speculate that stress, which contributes to depression, may be a significant component in this case because it has been shown to inhibit the hippocampus's ability to produce new neurons or nerve cells. Scholars are investigating potential correlations between depression and the slow synthesis of new neurons in the hippocampus. An intriguing antidepressant fact lends credence to this view. These drugs quickly increase the amount of neurotransmitters, which are chemical messengers in the brain. However, it usually takes a few weeks or more for patients to start feeling better. For a long time, experts have been perplexed as to why, if low neurotransmitter levels are the primary cause of depression, patients do not experience immediate relief from their symptoms when their levels rise. The explanation could be that mood doesn't go better until nerves develop new connections, which takes weeks. Antidepressants do, in fact, promote the proliferation and greater branching of nerve cells in the hippocampus, according to animal research. According to the notion, these drugs may actually be more beneficial for strengthening nerve cell connections, promoting the passage of information between neuronal circuits, and producing new neurons, a process known as neurogenesis. If so, it might be possible to create antidepressants that explicitly encourage neurogenesis in the hopes that patients would experience benefits more quickly than they do with existing treatments for depression. The brain's two hemispheres have mirrored versions of the areas displayed below. These structures also interlock. The drawing indicates a relative location rather than a specific location. Amygdala. The amygdala is a deep-seated brain structure linked to strong emotions like fear, rage, pleasure, grief, and sexual desire. The amygdala is activated when one recalls an emotionally charged memory, such as a terrifying scenario. When someone is unhappy or clinically depressed, their amygdala remains active even after they have recovered from their depression. The amygdala may actually grow larger as a result of this increase in activity. The basal ganglia are a related collection of deep-seated brain regions that are not shown in the illustration. They have connections to and interactions with regions located nearer the surface of the brain. They might assist with mobility and are involved in thinking, processing emotions, and memorization. People with depression have been shown in certain studies to have smaller basal ganglia as well as other anatomical alterations. Hippocampus. The processing of long-term memory is significantly influenced by the hippocampus. The proverb "once bitten, twice shy" may be explained by interactions between the hippocampus and the amygdala. When faced with an aggressive barking dog, this area of the brain registers fear, and the memory of that encounter may cause you to be cautious around dogs in the future. Some individuals with depression have smaller hippocampus, and evidence indicates that prolonged exposure to stress hormones may stunt neuronal growth in this region of the brain. Thanks so much for watching, and see you in the next video.